Hello everyone. So the topic that I've chosen today is plasma membrane. So before getting into the video, please do like, share, comment, subscribe to my channel. Thank you. So let's get started. Okay, plasma membrane. So what's a plasma membrane? A plasma membrane is a dynamic fluid structure which is actually found in the external boundary of cells, that is the outline, the boundary of cell. Now it will act like a selective permeable membrane. What does a selectively permeable membrane mean? What does it do? It actually regulates the molecular traffics that happen around the boundary of the cell. It will regulate those traffics that happens around that boundary. Now the plasma membrane will exhibit certain selective permeability. Now what's that selective permeability? The selective permeability is that it will allow very few or some of the solutes to cross across it when compared to others. It will allow some solutes to cross across it very easily, and some cannot even cross. That's what it does. That is selective permeability. It will only select few solutes that can cross across it easily. Now there were different models and different theories that were proposed. And in 1972, Jonathan Singer and Gart Nicholson, yes, these two proposed a model called fluid mosaic model. This fluid mosaic model was the most accepted model. Now what happened in this model? What's so special about this model? In this model, the membranes were viewed as a the membranes were used as a quasi-fluid structures. What's the quasi-fluid structures? Where the proteins were inserted in the lipid bilayers. So that's a quasi-lipid structures. Quasi-fluid structures, sorry. Where the proteins were inserted in the lipid bilayers. Plasma membrane is very simple. It consists of proteins and lipids, proteins and lipids, proteins and lipids. That's all. Now, what does this fluid mosaic model describe? What does it do? This involves two things. One is the mosaic arrangement and the next is the fluid movements. What's this mosaic arrangement? That is to see the protein embedded throughout this lipid layer. This lipid layer which consists of proteins. Now to see the arrangement of these proteins happened in this lipid layer, and the fluid movements of the same proteins and lipids that takes place. Okay, so what's this? Yes, this is a fluid mosaic model for the membrane structure. So it has an integral protein, peripheral protein and a phospholipid bilayer. Now what does this do? So the fluid mosaic model for the membrane structure, in this the fatty acyl chains in this lipid bilayer will form a fluid, that is in the hydrophobic region. Now these integral proteins will float in this lipid bilayer. Now both the proteins and the lipids are now free to move laterally in the plane of bilayer, but the movement of either from one phase of the bilayer to the other is restricted. Coming to the chemical constituents of plasma membrane, all plasma membranes, regardless of from what the source is, it consists of proteins as well as lipids. Now the ratio of that protein to lipid might vary depending upon the cell types. But let's go for an example. In an example, a human RBC has 43% of lipid and 49% of protein by weight, whereas a mouse liver cell has 54% of lipid and 46% of protein by weight. Now both the human as well as mouse cell definitely has carbohydrate present, and that carbohydrate might range between 5 to 10%, but there might be a range of carbohydrates present in each plasma membrane. But what's the most weightage? That's the protein and lipids. Now these carbohydrates bound either to protein as a consequence of glycoprotein or to lipids as a consequence of glycolipids. Now these carbohydrates are especially abundant in plasma membrane of eukaryotic cells, that is in humans, animal cells, when compared to prokaryotic cells. Okay, next, lipid bilayer. Now what is a lipid bilayer? The basic structure of the plasma membrane is the lipid bilayer. It actually composed of two leaflets, that is in the amphipathic lipid molecules which will have a polar head groups which will be in contact with the intra or extra aqueous phase, whereas their non-polar tails will face each other constituting the hydrophobic interior of the membrane. Now based on this, there are three classes of lipid molecules. Those are phospholipids, glycolipids and sterols. Okay, the structure that you can see there is a structure of a phospholipid, which consists of a hydrophilic head, which is also known as a polar head group, and it also consists of two straight lines, which is the hydrocarbon tails. Now, what is a phospholipid? A phospholipid is composed of four components: an alcohol, fatty acids, phosphate, and alcohol attached to that phosphate. The fatty acid components are hydrophobic, whereas the others are hydrophilic phospholipids. Now, talking about the phospholipids, there are different types of phospholipids. That is the glycerophospholipids, sphingophospholipid. Now, phospholipids, which are derived from glycerol, that is glycerol plus phospholipid, is glycerol phospholipid, and which is also known as phosphatidylglycerides. Now, what does this have? This have a glycerol, a fatty acids, phosphate, and one alcohol which is attached to this phosphate. Okay, for example, choline. So let's take an example. If the alcohol is choline, it is phosphatidylcholine. If the alcohol is serine, this is phosphatidylserine. The most commonly found phospholipid in the plasma membrane is the phosphatidylglyceride. Yes, that is the glycerophospholipids. Here in the sphingospholipids, this is as same, but the only difference is there, instead of glycerol, here we had sphingosine. That is sphingosine, fatty acids, phosphate, and an alcohol attached to it. So sphingomyelin is one of the abundant sphingospholipids. Now, in the plasma membrane of most of the animals consists of four types of phospholipids. That is phosphatidylcholine, phosphatidylserine, phosphonethanolamine, and sphingomyelin. Now, in the neutral pH, the polar head has no net charge, or sometimes they might have a negative charge, but having a positive. Now, talking about the sterols, the basic structure of a sterol is a steroid nucleus. It actually consists of four fused rings, that is, in which 3 with 6 carbons and 1 with 5 carbons. Now, it is planar and it actually has a rigid structure. Now, one of the most major sterol present is the cholesterol, which is found in the plasma membrane of animal cells. Now, the plasma membrane of plant actually lacks cholesterol, but instead of cholesterol, they have other types of sterols like stigmasterol and cytosterol. Now, even bacterial cell lacks cholesterol except mycoplasma. Now, coming to the glycolipids, the glycolipids contain carbohydrates, which can either be a monosaccharide or an oligosaccharide that are attached to the lipid. Now, these can either be derived from a glycerol or from a sphingosine. The simplest form of glycolipid is called as celebroside, whereas the complex glycolipid is a globoside, or which can also be known as ganglioside. Now, the cerebroside contains of a single sugar residue, which can be from the sugar, that is glucose and galactose, whereas the complex glycolipid, which is the globoside or ganglioside, contains oligosaccharides with one or more sialic acid. Okay, so now they are linked to the sugar chain. Now, the glycolipids are actually found on the outer leaflets, that is the outer part of a plasma membrane, where the carbohydrates portions are exposed on the cell surface. Okay, so that's all for today. Hope you guys like it. This is just a part 1.